answer, a never-ending story that is really quite evocative and, in my personal opinion, really represents the leitmotiv of an extraordinary scientific and human career. Thank you very much, Professor, for all what you did for myself and for many of us.
Thank you, Antonio. Good morning to all of you, and I'm really grateful to all of you for coming for this symposium. First, let me just thank the Institute for the hospitality and for the staff secretary help that they give to us, and the technology for all these slides and so on. I also would like just to thank our department for supporting the meeting, and the scientific and organizing committee, Beffa first, Riccardo, but also Alberto and Paola. Alberto and Paola did a very terrific work just to organize this, and thank you very much for this. Well, I would like just to speak about some of my early work on scientific career, at time just to brush up some hammer chords, if you like, of my early work, at time where not the sophisticated technologies were, and like PCR or NGS or omics technology. And on the first slide, yes, let's say, that was, I entered to medical course in University Bari in 1951, and that slide is just on 52 at the Institute of Normal Human Anatomy at University of Bari. I graduated in 57, also at the University of Bari, and just I had as a thesis, as thesis the attempt to experimentally reproduce the cardiac lesions that occurs in patients with the carcinoid syndrome, malignant carcinoid syndrome, which is a neuroendocrine tumor which have the neoplastic argentaffin cells of the normal gastrointestinal mucosa. So the attempt was just to produce liver damage in the attempt to eliminate the enzymes inactivating the 5-hydroxytryptamine, which at that time was isolated by Professor Vittorio Erspamer, a big pharmacologist, a great pharmacologist, and now it's, this substance is known as serotonin. So at that time, the 5-hydroxytryptamine was just experimentally studied, and in Bari, we had a series of experiments with this substance. The attempt was just to produce the carbon tetrachloride liver damage, cirrhosis in rats, and then given also the substance, the serotonin. So the results was that there was no cardiac lesions, fortunately, but as a serendipity, we found that the substance had a very profound protective activity on liver damage. So we published this work on laboratory investigation. And my mentor was Professor Luciano Fiore Donati, who later also helped me in my early career. So I enter in Institute of Pathological Anatomy, but in 1958, having got a fellowship from French government, I just moved to Paris at the Institut du Radium, la Fondation Curie, and that institute was directed by Raymond Latarget, who was a very profound investigator on the phage biology. I was assigned in the laboratory of genetics held by Georges Voudali. Georges Voudali was a very volcanic person, having a profound experience in endocrine system, in pathology and physiology of endocrine system. And I learned everything about how to use mouse, breeding mice, having all kind of genetic combination, and all kind of surgery on the mice, even the thymectomy. And last but not least, I also learned how to have the cell-free extract to generate virus material. And at that time, we were using the leukemic extract from leukemic organ from AKR mice in using the same technique that Ludwig Gross year before did. As you know, Ludwig Gross was the first to demonstrate the mouse leukemia virus in AKR leukemia, AKR mice leukemia, in this very fantastic and seminal work. So, I came back in Bari, starting mouse colony there, and having first studies on chemical carcinogenesis, mainly on perinatal carcinogenesis, and showing that neonatal age is particularly susceptible to chemical carcinogens, since they are just for the immaturity of cellular physiology and metabolic and also immunity. These studies were also shared with other Italian labs, like that of Della Porta, Beppe Della Porta in Milan, and Benedetto Terracini in Turin. And also in the States, there was in the Shubik laboratory in Chicago with Renzo Tomatis. Okay, back in Bari, we started also to work with viruses, with the Gross virus, but in 1961, having got a fellowship from the Deutsche Akademie der Wissenschaften in DDR, the Deutsche Democratic Republic, I moved to Berlin at the Institute for Krebsforschung in Berlin-Buch, which is Berlin East. And the director was Professor Graffi, Arnold Graffi, and here with his wife, Inge, and it's a carnival in the lab. Graffi was a very fantastic person, not only a very, very profound investigator in the chemical carcinogenesis, but also he had shown that from leukemic transplanted tumor, there was a virus, a leukemia virus, named also Graphivirus, who was able to induce myeloid leukemia. So that was characterized by the green color of the lymphoid organ, so it was a chloroleukemia. And Graffi was also a very cultured person, an artist, I would say, and besides written an important textbook on experimental Krebsforschung, he did also aquarelle, he was paintings, and he was also a composer. He did also compose some of the many Klavierstücke, is just like this. Back in Padova, we started to work with Graphivirus, with other colleagues, but in 1962, there was a meeting from the Italian Society of Cancerology, the SIC, just to recognize the work done in experimental cancerology. And in these slides, you will say the Umberto Veronesi and Beppe della Porta with Patrizia della Porta. And here is Gian Piero di Mallorca. Perhaps some of you will remember him. And Anna Maria, she was waiting, I think, from Fulvia at that time. Yeah. Okay. Let's go on. As I say, we were working with Graphivirus, which we obtained from Professor Graffi, and we, together with Luciano Fiore Donati, we did studies showing that the type of leukemia was very much dependent on the genetic background of the mice. So being possible to induce lymphatic leukemia or myeloid leukemia, and even in the same animals, in the same animals, we could induce a lymphatic transformation, lymphatic cell transformation in the thymus and in the rest in the chloroleukemia. These results were later confirmed some more, about 20 years later, by a Canadian group showing that Graphivirus is a multipotential virus being possible to induce different type of leukemia in the host cells. Now, at that time, there was a great influx on the immunology. And the studies of a young investigator in UK, which is Jacques Miller, he was working at Pollard's Wood near London, and he was working also on the leukemia in AKR mice. And he showed that by making neonatal thymectomy, these animals did not induce no more leukemia, but more important, they got a wasting syndrome. So in other words, they were dying with a completely deprived lymphoid tissue. And that was the starting of the recognizing that in mammalian organs, there were the cells which were very important for the immunity, the so-called cellular immunity. That was the T cell, the first experiment showing that there was T cells. And here, Jacques and Margaret Miller in Ischia in summertime. We were in vacation together, and here Jacques Miller who got the Lasker Prize some years ago. Now, these studies were important just to, for us to show that the immunity in virus-induced leukemia was a very peculiar situation, since by injecting the virus as a neonatal age, we could induce kind of immunological tolerance. And that was a very, starting observation for a series of experiments we did later. But before, I must say that in the first meeting we had in Padova, when we moved in 1965, was on germinal center of lymphatic tissue. That's Ricardo showed the very complicated, very important molecular physiology of germinal centers with the arrangement of genes of B cells. But in 68, we had this meeting together with the Oak Ridge group, Oak Ridge in the States group, collecting all the immunologists at that time, and there was Jacques Miller, of course, but also Max Cooper and Bob Good and others. And that was very interesting meetings. Of course, there was mainly on the morphological and functional studies on the germinal centers. And we were able also to provide to the participant the original, the copy of the original paper by Fabrizio di Acquapendente, who was the founder of the Bursa, from which the cells were named B cells. And so that was particularly appreciated. So we were in Padova. We moved in Padova in 65 with Fiore Donati and Pennelli, Natale Pennelli, a colleague of us, and Tridente, and Giuseppe Tridente. We were following our boss, our professor, the director of our institute, Professor Vizzi, who got the chair in University of Padova. And the first time were very heavy for us as duty, just because to organize all the work, but also from the family work, because we had the two daughters. Fulvia was 3 years old, and Maria Alessandra 4 months. And Anna Maria, my God, who I was married in 61, just after my coming back from Berlin. Anna Maria had to change her work from the archaeology of the Magna Grecia to the archaeology to the Paleoveneti. So that was also very important for her. What else? I was just jumping a little bit because I make more. As our work, we were also working with the colleague in hematology field, and in 1969, as Rudiger, as just called, we found, together with other colleagues, the Italian Association Against Leukemia, the AIL. And here in these slides, we were the foundation of the association with the President of the Republic, this Giovanni Leone, and other colleagues. You may recognize here Felice Gavosto here, and Fiore Donati here. And the president was Fabrizio Alleata di Montereale. Fabrizio Alleata di Montereale, who was married the Ianna Cini, the daughter of the Count Vittorio Cini, and that was very instrumental for how other work, since Ianna Cini and Fabrizio Alleata were very much friends of Albert Sabin. And so we were able, in this way, just to organize the first meeting in Venice for the comparative leukemia, as Rudiger told. And, this is the 50 years of AIL in 2019, and with Santetura, Rudiger is there, of course. So I told this is the first comparative leukemia meeting in 1971 in Padua and in Venice. Slides is with Albert Sabin, and there was in the same magazine, there was also a very, very complete article written by Barbara Colleton, and showing that some of the participants, like you see, young Dr. Gallo here, showing that the reverse transcriptase was important for the leukemia cell transformation, and George Todaro, also back from Padova, but also a young guy that was just coming back from the from Malaysia with helping there, and also the Guy d'été, very elegant, in a gondola. But also I will also show you some of the other participants, like Tony Epstein, and here is Ray Dutcher at the time was the Secretary General of the association, Anna Maria, and here is Nekama Arangera from Israel. Henry Kaplan, who was receiving a prize from the Leukemia Foundation of America, and Joe Barchenal, you remember, and with Nicole Mueller-Bevart, that she was the editor for the leukemia, as perhaps Rudiger knows. And here is Danny Bolognesi, Bob, do you remember? And Giampiero di Mallorca, Guy d'été, was it was Schlom, Jeffrey Schlom is here. Yeah. Okay. We had a concert in Villa Contarini in Piazzolo del Brenta, very nice villa, and with Simone, Vittorio Simone, having as a conductor. And here there is a Simone giving an autograph to Albert Sabin, and Anna Maria and Fulvia are also there. We had 3 more comparative leukemia meeting in 91 and in 2001. And as you remember, Rüdiger had other 2 in Mannheim and Heidelberg, and Beppe Saglio in 2013 is in Turin, and next will be in Bologna this September. So another period for me important was that in 73 I was at NIH with the Fogarty Fellowship, and I was working in the Bob Hubner branch in viral oncology, and particularly in the Tadao Aoki laboratory that was in building 41, EBEFA, in your building. At that time was just a prototype of the SBL3 for tumor virus. Is very important. And together in the same building, but a different suite, we were also the Peter Fischinger and other Skolnick and Park and Skolnick. Tadao Aoki was a very tough Japanese boss, and it was an electron microscopy, but was very attent to hierarchical power for the old Japanese, the poor Japanese colleagues that we were working in the same suite. To me, it was very, very kind, and I was free to just to do some work in my own line of research, and so I was able also to continue to work with the tolerance story, and we were able to publish a very important, for me, very important work with the Maloney sarcoma virus, showing that if the Maloney sarcoma was injected in AKR mice with endogenous gross virus, you shift for phenotypic mixing the virus, the Maloney sarcoma. So there was no regression, no spontaneous regression of the tumor, but it was just continuously growing, just for the possibility of, again, tolerance, immunological tolerance to the newly formed gross leukemia and sarcoma virus. So, and that was, this paper was also worked with Peter Fischinger, who was very important in having all the neutralization of the viral things. Concluding the story of the tolerance, we had in Padova a very important group with Dino Collavo. Dino Collavo was very instrumental to define the cytotoxic T cells, virus specific, and he was taking expertise in this kind of immunology, working also in the Chester Beatty, Tony Davis lab, and later on at Ludwig Institute in Lausanne with Cerottini and Brunner. So Dino, with Franca Ronchese, you visited yesterday. She is now in Wellington, New Zealand, as professor of immunology, but also three sparkling investigators, Emma D'Andrea, Anita De Rossi, and Paola Zanovello. We were just working that with Dino. And that was a very important paper showing that in these MOV-13 mice were a line of mice obtained by and produced by Rudolf Jenisch, when at that time still in Hamburg, and with endogenized Moloni virus, Moloni leukemia virus. That was that Rudy was just injecting embryo with the Moloni leukemia virus, and in this way, these animals had the provirus, the DNA provirus, integrated in all the somatic cells. So they were just as a completely endogenous virus as Moloni. And in this particular line, the expression of the virus was also before the birth of the animals. So it was completely antigen release before the birth. And so there was a completely tolerance, and that was also shown by the completely absence of the cytotoxic T cells specific for the virus. And there was a very clear demonstration of that the tolerance was very important in having the development of leukemia in animals. And this paper received also the congratulation of the reviewer, that's to Franca Ronchese, I would say. Okay, that story of the tolerance seems not only kind of just experimental things that will be just demonstrated in the lab, but perhaps it has also some importance, since now there is the possibility that tolerance, immunological tolerance, is existing in bat. You know that bats are now very important animal species for the spillover of the many diseases, virus-induced diseases. And there is here a series of papers showing that in bats there are a lot of endogenous viruses, not only retrovirus, but also viruses like the SARS-CoV-2 and others. So it's important that perhaps the bats may just release this virus without being affected by the virus in the same. In the meanwhile, we had a very good support for our work from the CNR, Consiglio Nazionale delle Ricerche, with a special project on oncology. There was some important support, financial support of our research, and here is one of the scientific council for this project, with Umberto Veronesi here, and Rossella Silvestrini, and Alberto Forneso. You may also recognize Beppe Della Porta, Giancarlo Vecchio, and Felice Gavosto. Oh my gosh. My God. Okay. What else? I would say that there is also Paolo Comoglio and others, okay. And that was very important project from the CNR. But also a great support we also had from the AIRC. Federico just mentioned yesterday about our support from the AIRC. And I would say that besides grants to the Institute of Tumor in Milan and Institute of Regina Elena in Rome, one of the first grants given by the ARC was that in Padova in 1970, and we received that. So thank you also to ARC. In meantime, the recent story is known to all of you. I will not show the work. That was the IL-2 discovery, and then HTLV-1, and then HIV by the laboratory of Professor Gallo. And the story of this discovery are well documented in this book that also document the copy of letters and documents showing the story of this discovery. Of course, there is an English edition of this book. Bob, you remember it? Yes. Okay, so I will not go through this. We also had some work done, and that at the beginning was from the generosity of the Professor Gallo, the Bob, because he gave us reagents, cells, and so we started to work also first with HTLV, and then, as I will show you, on HIV. And we started to do the epidemiology in our regions, and at that time, HIV was named HTLV-3 in the early times, before having the new nomenclature. And Anita Di Rossi started with other colleagues, Anna Rosa Del Mistro, to make this kind of epidemiology survey. And we also had, with Anna Rosa Del Mistro and Anna Aldovini, these studies doing immortalization of human T lymphocytes by HTLV with kinetics of virus integration and expression. So that was our very early studies. But then Anita conducted all kind of studies with HIV, and as she showed the first day of the meeting, the very, very important groups on pediatric work on HIV. And she had also the possibility to have these international groups of PENTA together with the pediatrician in Padova, Carlo Giaquinto. So that was a very important group. We still continue to work on that. And the studies on the chromosome, by the way, were later on also confirmed later, some 20 years after our studies done with Daniela Saggioro and Anna Rosa Del Mistro and others here. And we found that there was some chromosome alteration by HTLV-1, and later on was also confirmed by T. Zhang in States, And you can see here, we're very, very friends, coming back from a meeting in Japan. Alberto Amadori, the other way, he was working mainly on B-cell activation in HIV-infected person. And he also did very important work showing that in these patients there was also a B-cell dysfunction and showing this hypergamma globulina in patients and in kind of very different work. Lab meeting in Bob. We were in many of these lab meetings in different, that was at NIH before Bob did move to Baltimore. It was in '86, and you can see here Bob with Luc Montagnier, and there was Daniel Zaguri, Maurice Hillman, and but also, oh my God, Michael Feldman, who was, I was working with Michael Feldman at Weizsäcker Institute. I've forgotten to say that I was in Israel in 1963, and this was my mentor in Israel. And Danny and Hazeltine. Okay, and also there are some more people. So these lab meetings were very, very important for discussing, having freely with all together. And Bob was very successful in doing all these things. Thank you again. But also in Europe, we had very important leukemia meeting, and Wolfnet was the organizer of this Vilsede, and Rudiger also said about this. You may see here some, this is, would say, Max Essex here, Peter Fischinger, Arsène Berny, and here there is Robin, I think, and Jean-Paul Lévy and Harald Zuhausen, Riccardo, and Cesare Peschler. Yeah? And Carla? What is it? Yeah? Okay. And here is Peter Duisberg with George van der Bude. This is the Peter daughter here. So also these meetings were very, very important to, for our leukemia groups meeting. Now, in 1991, there was the International Conference on AIDS. That was just to recognize the big effort of Giovanni Rossi in Rome to organize a special program on the AIDS studies. And this program was very, very important for us just to obtain support for research, and the program was also able to bring in Italy also young investigator, Italian investigator working outside, for instance, in the States, and Guido Poli was just coming back. But also Barbara Enzoli was also coming back, isn't it? She was in the Bob lab, and she came also back and working in Rome at Istituto Superiore di Sanità. So I will just showing that in our place we did form also the Department of Scienze Oncologiche e Chirurgiche in 1966. That was the first groups of our department in Padova. And lastly, I would just to show you some slides of the first meeting of this series, the virus, genes, and cancer. We had in 2007 here. And that was also a coincidence with the birthday of Professor Gallo. And you may see here a very nice okay, I will. Bob is just receiving a gift, a sculpture, a glass sculpture from Moretti, showing that DNA helix. And here you see Robin. She is a wife of Guy Dété, Bob and Mary Jane, with Claudio Basilico, and Peter and Gunnar Bieberfeld, Bob with Ira Fustenberg, and Emma D'Andrea, Robin, and Stu Aronson, Donna D'Agostino, and here there is Eva and George Klein. Again, Robin, Arsène Berny, Rudiger, and Rupert, and Dino and Antonio Rosato. And here Volker Erfle, our very good friends. I would like just that all of us will send to Volker our best wishes. We hope that he will be back with us in soon. And Paola here, and Alberto, and Daniel Zaguri. Again, oh, sorry, that's important. Carlo with doctor. And Robin Stewart, and this Ray Wagner, and Marjorie Robert Guroff, and Anna Aldovini, and George Pavlakis. Okay, I would say, sorry, no, that's important. That's very important. Vestergaard. That is the GVN. This was an initiative that Bob, that was founded in 2011 with Bill Hall and Rainer Kurt. And I had the honor to be the first Italian coordinator of the group, because Bob asked me to do that. But then the coordination passed to Umberto Bertazzoni, and then now to Franco Bonaguro. He's now the coordinator of Italian groups. And now, very recent news here. There is a step down of Professor Gallo, who became emeritus professor at University of Baltimore, but it is not like the 86-year-old biologist is retiring, so he will continue still to work with us. So we hope very much. So I'm sorry for having this broken English. Is not like the king. Oh, well, yes. I had also this very, very late. That's for you. One more. Thank you. And there was not a King's English like my grandson Francesco said yesterday, but a very, very broken English. Okay, thank you very much for all of you for your friendship. Thank you.
Well, what to say? Thank you very much for this simply fantastic journey in a science that has really produced enormous results, has evolved, and it's a really pleasure to say that at least with many of the persons Professor Chieco-Bianchi mentioned, and I had the pleasure to work, and I still had the possibility to work with some original models like the Moloni sarcoma and leukemia virus. I learned from Professor Chieco-Bianchi the thymectomy. I learned from my other mentors, Professor Collavo, Professor Zanovello, how to make CTL clones, how to cultivate and to study immunology. Really a fantastic and incredible story. There were some closing remarks actually by Riccardo Della Favara, Genoveffa, Franchini, Alberto Maduri, Paolo Zanovello. The organizers say that we can close, but I'd like to invite at least Riccardo and Veffa just to give and to say the last few words. Thank you very much.
Okay. Wasn't prepared for this. What can be added after 2 days? What can be added to the remarkable story of Gino Chieco-Bianchi? I just say the following. If one has to depict the ideal scenario in our career is to have a successful lab with a lot of discoveries, to have created a school of not only good people, but people that keep on remembering you and respecting you, and having a great family that celebrates you at the end of your career. I think a lot of us cannot accomplish all this. It's a dream. But Gino did. So that's what I saw today. And so congratulations. Really.
On a less serious note, I would like to say the following, that this meeting has epitomized not only the legacy of Gino, but also has put together people and exposed them to a virus, CoV, which is really, which is the SARS-CoV-2. So, but thanks God, if we have to believe Buonaguro, maybe virus mimicry will decrease our chances of getting cancer. So I would say that not everything comes in a bad way. You can perhaps have also see some positive in everything. But on a more serious note, it was really a fantastic example, I think, of how cooperation and friendship together can make science progress, and how the legacy of people like Bob, like you, and can really put the people together. Even Riccardo and I became more friendly recently. So I think collaborating. So I think this is really amazing.
Let's not go too far.
Which Palazzo? No, Franchetti. No, it's very different. But Very different, very different. By the way, this Franchetti Palace, there is the owner of this. I know him. He's in Washington, ex-owner. His grandfather is the one that fixed the how is it called, the Palazzo, uno dei Palazzo dei Dogi, and it was his grandfather. And he sold it, but when he saw that I was going in, he says, "Hey, you're going to my old house." I said, "Wow." By the way, he's very rich, as you can imagine, because he sold a lot of properties in Venice. Anyway, thank you to everybody for really giving a great contribution, and also it was a very good spirit present in this meeting, I think. Thank you to Paolo Alberto for really doing most of the work, and to you also for helping a lot. Thank you.